Hello friends, today we are going to study about protein function under which reversible binding of a protein to a ligand part 3. Now the hemoglobin's oxygen binding curve is sigmoidal. In the last video, we studied about myoglobin's oxygen binding curve and we found that it is hyperbola. Here in hemoglobin, its oxygen binding curve is sigmoidal. Hemoglobin is a multi-subunit protein. How many subunit it has? 4. Now for a protein, so that means it has 4 binding sites for its ligand. And what is the ligand for hemoglobin? The oxygen. So it can bind 4 oxygen because it has 4 binding sites, each on each subunit. Now for a protein having n binding sites, the equation is the same equation as we studied in the last video, that is P plus L reversibly equal to PL. Here also it is the same, only we have added this n, that is n number of ligands can bind because the binding sites are also more. So the Ka, that is association constant, will become the same as in myoglobin. Only this extra n will be there because in each protein n number of ligands can be bind. There in myoglobin, per protein one ligand because myoglobin is a single subunit. But here hemoglobin is a multi-subunit. That means per protein multiple ligand can bind. Here it is 4. How it will affect the theta equation? You remember this was only the theta equation. In case of myoglobin also, here only n is added extra. Now this in term of oxygen we can write it as, because oxygen in terms of partial pressure, so Kd is that concentration of ligand when the saturation is 50%. I hope you remember the equation from the last video. Those who have not seen that video, for detailed explanation, you can check that video once. Now, similarly, as we plot the graph between theta and partial pressure of oxygen, we obtained hyperbola. But for hemoglobin, by plotting the same graph, we are getting this sigmoid. You can see this. This is a sigmoidal curve. Why here YO2 written? In the last video only I said, in some book you can find theta. In Leninger specifically, you can find this theta. In Voigt, you can find to be labeled the fraction as YO2. So this curve, the green one, which I'm making it green now, is of myoglobin, which we already studied. And this red one is a sigmoidal curve for hemoglobin. Now you can see there is a difference between both the curve. And what that difference is? In the sigmoidal curve, it is not as steep as myoglobin. In the beginning, you can see there is like this, a slant is there. And then we can find the steep, and then again it became static. The reason for being static is clear, that all the binding sites are now occupied. That's why increase in concentration of the ligand further will not affect the fraction, of course. Why the graph is not as steep as myoglobin in the beginning? The reason is such type of sigmoidal curve is produced because of cooperative effect. What do we mean by cooperative effect? Cooperative effect means the binding of one ligand will affect the binding of another ligand. If you remember from the myoglobin video, not going much into detail, myoglobin is a single subunit protein. So the ligand that will bind to these subunits, they are all independent. That why? Because the ligand binding to one protein cannot affect the binding of ligand to another protein. So all the ligands are binding individually. But if we see the structure of hemoglobin, in that case, in one protein only, we have 4 binding sites. 4 oxygen or 4 ligand can bind. So the binding of the first ligand will affect the binding of the second ligand. The binding of second ligand will affect the binding of third. The binding of third will affect the binding of fourth. And how it affect? It affect positively. That means the binding of first ligand will increase the affinity for the binding of second ligand. The binding of second ligand will further increase the affinity for the binding of third ligand. So like that it works. So that type of association or effect is known as cooperative effect. When the first ligand is binding, for it, there is no cooperation, no cooperative effect because no other ligand has bind. So the first ligand which will bind, it is binding independently. So that's why the graph has been shifted a little. It is not that steep. It is slow. When the first ligand has been bind, now it will affect the binding of the second will affect the binding of the third. So now the graph is started rising as the concentration of ligand is increasing. And finally, when all the four site has been occupied of all the hemoglobin molecules, it will become static. Clear? So now it is clear why the graph of myoglobin is hyperbola and why the graph for hemoglobin is sigmoidal. All the hyperbola curve is a result of individual binding of ligand and all the sigmoidal curve represents the cooperative effect. Now the P50 for myoglobin, it is 2.8 torr. P50 means when the protein will be half saturated. So the P50 for myoglobin is 2.8 torr. The partial pressure of oxygen in arterial blood is 100 torr and in venous blood it is 30 torr. In the graph also you can see this is venous pressure, this point is 30. This is arterial pressure, here it is 100. So the concentration of oxygen in the arteries is 100 torr and in the venous it is 30 torr. That means myoglobin will always remain fully saturated whether it is an arteries or it is in veins. So that's why myoglobin is better known for oxygen storage. That's because in the venous and artery, whether, as you all know, in the arteries, oxygenated blood flows, in the venous, deoxygenated blood flows. But in the venous also, the concentration is 30 torr, which is much higher than P50 of myoglobin, which is just 2.8 torr. So whether we talk about arterial blood or we talk about the venous blood, myoglobin will always remain fully saturated. Hence, it functions better for oxygen storage. But if we say about hemoglobin, the P50 for hemoglobin is 26 torr. That means in case of arterial blood, when the pressure is 100 torr, it will remain fully saturated. But in the venous blood, when the pressure is 30 torr, it will be only half saturated. Whereas in myoglobin, it is fully saturated in both the cases, but in hemoglobin, in venous blood, it will be half saturated. Now, this difference is important to release the oxygen. The difference in the concentration of oxygen in the arteries and the veins can be sensed by hemoglobin and not by myoglobin. Because for myoglobin, both the concentration is way much higher compared to its P50. So for myoglobin, it is very difficult to sense the venous and the arterial blood. But it is easy for hemoglobin to sense the venous and the arterial blood because in arteries it will remain fully saturated, whereas in venous it will remain only half saturated because the concentration in venous is 30 torr and hence it will remain half saturated because the P50 of hemoglobin is 26 torr. And this difference is needed to release the oxygen in the body. Hence, hemoglobin is designed to carry oxygen and myoglobin is designed to store oxygen. Now, we obtained the theta equation. Now, after rearranging this, we can get taking log both sides. This equation is known as Hill equation. And when we plot a graph between log L and log theta upon 1 minus theta, this will be known as Hill plot. In the Hill plot, we'll get a slope which we called as N. So what is N? The number of ligand binding site. But experimentally, it is actually not the number of ligand binding site. It is the measure of degree of cooperativity. And hence, because experimentally it is the measure of degree of cooperativity, so we do not write as N. The N is only written as NH. That is Hill coefficient. NH is equal to 1. That means no cooperation. NH greater than 1, positive cooperation. NH less than 1, negative cooperation. Now let's see the graph. This center straight slope here, N is equals to 1. That is myoglobin, no cooperation. This sigmoidal is of hemoglobin. Now here, this part, if we extend like this, then it becomes parallel to 1. That means here also NH is equal to 1, which shows the low affinity. Why? Because this is the binding of first oxygen molecule where no cooperation is seen because that is the first oxygen. So among all the ligands, all the oxygen will fight for that first position. On the upper side, if here also after extending this line, this is also parallel to this red myoglobin line. So here also NH is equals to 1, but here it is high affinity. That is the binding of the last oxygen molecule. Here also NH is equals to 1. That means no cooperation, but with high affinity. Why? Because now the fourth molecule also bind. So after the binding of the fourth molecule, what else cooperativity it will show? There is no other place for binding of any fifth molecule. So there also it will be no cooperation, but here the affinity is high because this is the binding of the last molecule. And for hemoglobin, this NH value is between 2.8 and 3, which shows that hemoglobin is highly cooperative. So for hemoglobin, the Hill coefficient is between 2.8 and 3, which shows that hemoglobin is highly cooperative. So this is the Hill equation for both hemoglobin as well as for myoglobin. Now one question from my end for you all, that is calculate the fractional saturation of hemoglobin at partial oxygen of 50 torr and when N is equals to 3. Do let me know in the comment section. If you find the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.